karakia. Ka tika kia taki karakia kia pai ai te haere o tā tātou hui. Nā reire kia taunga manaakitanga a te mea ngaro, ki runga ki tēnā o tātou. Kia māhea te hua mā kihi. Kia toi te kupu, toi te mana, toi te aroha, toi te reo Māori, kia tūturu ka whakamaua kia te inga. Hau e hui e tā i ki. Kia ora tātou. Kai a koe Maui.
Kia ora. Nō reira, tēnā koe Holden. Nā noe tuku ngā karakia he wāwahi kōrero mō tēnei ata. Kia ora koutou katoa. Hiani i tēnei ata. So welcome to this Whāki webinar on eDNA, Exploring Māori Data Governance and Sovereignty. Really excited to have so many people here, and I'll just put up the slide so that you can kind of see what the agenda is and for the webinar this morning. Let me just get this up. Okay. Cool. So I'm going to start with introductions of the speakers. We'll introduce 3 projects that have all got sort of overlapping interests in this space. One's a National DNA Reference Library project. One's an environmental project around marine fishes and freshwater insects, and the MFE Stygofauna project. And then try and give a little bit of a kind of context for people thinking about what does it mean to be involved in DNA sequencing, and particularly barcoding activities that are associated with these projects, and some sort of work that's been done to just explore what Māori data governance looks like. So we're intending to do a bit of a presentation, have some space for a Q&A. That will take the first hour, have a short break, and then do some breakout groups with those of you that want to hang around for the second part of it as well. So just by way of introduction, I'll just stop sharing this now. Ko mā tātua te waka, ko whakatohia te iwi, ko ōpape te marae, ko Maui Hudson tuku ingoa. So I'm the director of the Te Kotai Research Institute at the University of Waikato, and I've been one of the team members associated with the National DNA Reference Library. Had a component in there alongside my colleague Rogena Sterling, just looking at some of the different ways in which people are thinking about governance in relation to different genetic databases. So kia ora. And I'll just pass it around to Holden in the first instance, and then the other panelists as well.
Kia ora tātou. Ko taranaki tōku maunga, ko waitara tōku awa, ko te upoko o te whenua, me te tata o te pō, waiwhetū, me pipitea o oku marae, ko Ngāti Maru Wharanui, me te atiawa taranaki whānui ki te upoko o te ika, oku iwi, ka mutu ko Holden Hohaia, tōku ingoa, kia ora tātou. My name's Holden. I'm the co-lead for this mahi with Manpreet. Kia ora tātou.
Nice segue to Manpreet.
Kia ora koutou, ko Manpreet Dhami Takawango. I'm a senior scientist at Manaaki Whenua Landia Research and helping Holden with the National Science Challenge project in exploring how we might have Te Tiriti values built into every part of that process of generating a national resource. Yeah, that's me.
Sure, thanks. Aimee.
Morning, everybody. My name's Aimee from the Reis, and I'm based at Massey University alongside Libby Liggins. And my involvement is making up molecular libraries, specifically for groundwater stygofauna, and also part of that's with MFE, and then also part of MB EnviroLink project that's doing libraries for marine fish and freshwater insects. Thanks.
Kia ora. And hopefully Libby will be joining us at some point. She has a little baby, so these sometimes there's things that get in the way, so we'll just roll with it. Okay, so back to the slides. Kia ora. Manpreet, Holden.
I think this is me, Manpreet. Yes. So this is essentially just an overview of what we've been brought on to produce. And I think it's important to just stress here that we're not actually tasked with producing an eDNA reference library. What we've been tasked with doing is to produce some advice on how we might go about producing a library. So the product, if you like, is advice to the government on how you might go about doing this in a Te Tiriti-founded way. So that's the key point there. Manpreet, did you want to add anything?
I think really it's just a reflection on all the ongoing work that's happening, and in the past, the work that has happened around these ideas, and we're just kind of trying to meld them into developing a national resource for biodiversity monitoring. And so it's a bit different, but at the same time, we are engaging and trying to gather as much information as we can so we can, inform that the business case for this database moving forward for the government.
You keep going, you're on a roll.
Okay. This is just to highlight the working groups. As part of the project, we've been doing a few activities, and some of you have obviously come to the wānanga series that Sarah has been helping arrange and organize. But I just want to reflect on the two major pieces that are currently ongoing, along with the work that Maui and Rogena, and I think Libby's involved with that as well, are also doing around gathering this information and helping us make the best informed case in this point in time that we can. The project working group have been, it was initially set up at the start of the program around February, and we've met, and that's where the initial thinking came from. Okay, what are the open gaps for within the biodiversity conservation space around these ideas? And this is just a list of different project group members from around Aotearoa, and from that we developed the wānanga series to highlight the topics where we wanted to think more before committing to one way or another, and that's what we've basically been doing. And I think that's pretty much, it would be fair to say this is very much still work in progress, even in developing that advice piece, and workshops like this one and also the wānanga series have just been excellent at bringing people into the tent and opening our minds as well to everything that's ongoing that we can learn from. And I think I'll plug in our next wānanga, which is I think early next week, so in Monday next week, and that will be looking at cross-project fertilization, so looking at a lot of actually international projects that have been dealing with some of these issues and a couple of our Aotearoa specific projects as well. So I think I'll stop here.
Aimee, I think, can you do this? Libby's not on.
I'm here. I just made it.
Oh, here's Libby.
Great. Apologies for the technical difficulty, but I'm here now, so I'll share these slides with you that Aimee and I have been working on. So Aimee and I are researchers.
Just do a quick intro to everyone.
Oh, okay, sorry, I wasn't sure if they'd gone already. Kia ora. My name is Libby Liggins. I'm a researcher at Massey University, and together with Aimee van der Reis, who's going to be presenting some of the slides subsequently, we're nominally leading a couple of projects out of Massey University around generating DNA reference databases that we hope are fit for purpose, both scientifically and culturally. But of course, we don't know what that means just yet. So our approach is to be learning as we go. So using experiential learning where we make a start, but include some time to pause, reflect, and redirect when necessary. So the first project is the one you can see in the slide there. It's EnviroLink Tools project. And these are regional council-driven funding schemes that provide research support to regional councils in the environmental management space. So our projects are a collaboration between several regional councils, DOC, museums, universities, and other research entities, and it's motivated by the increased use of environmental DNA or eDNA approaches by environmental and resource managers globally. To effectively use an eDNA monitoring approach requires being able to associate the DNA collected from the environment with a species of known taxon identity. So that's the purpose of a DNA reference library, which we're talking about today, of course. So our grand goal is to provide the DNA sequence information that's essential for being able to detect certain species in an eDNA sample taken from Aotearoa, New Zealand, therefore delivering a better tool to resource managers and communities that's more sensitive and able to detect our rare and threatened species of interest, indicator taxa for ecosystem health, and overall ecosystem change. So our focus for this project is actually on marine fishes and freshwater insects, and that's been informed by the current needs of councils that have been communicated to us, as well as correspondence with Department of Conservation, and also an increased awareness and desire to understand linkages between our freshwater and marine systems through really important estuarine environments. So Maui, if you skip to the next slide, our approach in doing this project is to generate data from existing, expertly identified and vouchered specimens that are held in national collections where possible. So for some species, of course, we will also be undertaking fresh collections where they don't already exist, and this will be done in collaboration with council staff, DOC staff, and mana whenua. So these will then be identified and accessioned into those national collections, so a resource that's being held and curated and looked after by our taxonomist and museum teams. So for these specimens, we're looking to generate short DNA barcodes in this project, and these are commonly used in eDNA studies to identify species of these taxonomic groups. If you jump to the next slide, Maui. Once the DNA sequences have been generated, they are added to a repository called the Genomics Observatory's Metadatabase, or GEO. And this is a repository that other DNA reference sequencing projects have used very successfully. The repository can hold all the important specimen information, so all of the metadata, as well as the sequences, and can be kept private, but in a format that's interoperable with other data repositories that store this kind of DNA reference information. The metadata can be hosted here. The metadata that can be hosted here includes all the important ones that helps it be interoperable with other sort of biodiversity collections. But it also has some fields that can be co-opted to hold Indigenous metadata or metadata relevant to Māori contexts. There's also the opportunity to develop new metadata fields, and that's something that we're hoping to do a little bit of in this project. Importantly, this repository is also completely interoperable with the Local Context Hub, which operationalizes the local context system of biocultural and cultural notices and labels. And this is really important, especially in the case of existing collections, where we're aware mana whenua may not have been involved in the collection event and may not have knowledge of these collections actually existing. So we're talking about collections that may be 30, 40 years old. So in these cases, we're working with the museum partners to explicitly disclose that there is incomplete information with regard to the Māori provenance of these collections, and attribution is incomplete using institution notices. And biocultural notices are applied to the specimens and all data that derive from them, making explicit the rights of mana whenua to define the use of these data from the biodiversity and genetic resources associated with their traditional lands and borders. So the second project I'll just go over really quickly because it's quite similar. This is directly funded by the Ministry for Environment and has a dual focus on obtaining and sequencing voucher specimens, similar to the previously described project, but also is around facilitating greater cultural engagement around molecular techniques and DNA reference libraries. And this has been led by Maui and Rogena, and it includes this webinar today. So this project, again, has several partners and crucially relies on the expertise of scientists at ESR and NIWA, who are the real stygofauna and freshwater crustacean specialists. The project focuses on groundwater systems, which are often neglected due to their out-of-sight nature and the logistical challenges in actually sampling them. But these hold really unique biodiversity, of course, that is under threat from extraction and pollution. So in this context, an eDNA approach provides a practical option to monitor these groundwater ecosystems from both an ecosystem health perspective as well as a biodiversity perspective. So the next slide, Maui, shows us that the approach is really similar to the EnviroLink one. The only difference being that here we're looking to generate DNA sequences that we're terming mega barcodes, which are much longer than typical DNA barcodes, but really serve the same function and that they can help us identify species. More on that later in the webinar. Next slide, Maui. And again, the subsequent treatment of this data is very similar to the other project. It's been held in Geo privately until project completion, when we'll be looking to the outcome of kōrero, such as what we're having today, as to what best practice looks like in Aotearoa for treatment and the governance of these kinds of DNA reference databases. Thanks, everyone. Apologies for the late arrival, probably mostly to the facilitators who may have been stressed out. And thank you, Aimee, for offering to step in, obviously. Look forward to chatting today. Thanks.
Thanks, Libby. So yeah, that's a sort of a bit of an outline of these 3 projects that are coming together because they've got sort of similar interests, and all of them have a similar interest in how Māori data governance and issues around Māori data sovereignty should be playing out. We've sort of heard a little bit of that. I think part of the background information around this is really thinking about when sort of dealing in these spaces with DNA and we think about genetics, often, unless you're an expert, and I'm not an expert, you think about them all as pretty much the same. And the genetic information being generated is the same happening in all the places. And I think that is something which is clear from this table which Aimee's put together. We've been trying to demonstrate how some information is useful for a whole lot of different purposes, but some information is probably only confined to the activity it's being collected for. And just showing here different things that you might hear today around a barcode or DNA barcode, mega barcode, the reduced genome skimming, and then a whole genome. And on the left-hand side, it gives you an idea about the kind of the different size and information being collected, and what that then can be used for. And so a lot of the work that will be, or the projects that are being talked about today are dealing with things at a barcode end of the scale. And so it can be useful for species ID. It doesn't need really high quality DNA. And it's unlikely to be able to be used for other things. And so I think that's something that just need to keep in mind when we're thinking about some of the questions that will come up later as well. And just saying, feel free to throw any questions in the chat as we're going along. We're going to have a Q&A session afterwards. And just to give you an idea about like the increase in the amount of activity that's happening around this sort of barcoding and mega barcoding space. There's been a huge increase in the number of publications based on a huge increase in the number of activity that's happening there. And so seeing Libby talk about councils becoming interested in using it as a part of their monitoring activities, and the push towards having a national reference library as something that can make things consistent as well. This is one of the things that we then see is that there's different ways in which genes are sequenced. And there's difference between sort of full and partial reads, the coverage, so how much of the gene is being sequenced and to what depth it's being sequenced. And so that makes a difference and sort of sets up a difference between what is the information that's getting through the barcode exercise and then its relationship with a reference genome. And so I think these are things that we can certainly ask questions from Manpreet and others and the people that have more technical expertise. But I think understanding that is important because it shapes the way you think about how that information can be reused in different ways, and therefore what sort of maybe controls you might want to place around different sorts of information. The other thing that I think is probably worthwhile thinking about is where the information ends up and what sort of metadata sits alongside it. So here you can see, kind of a sequence itself, or some of the base pairs that are sitting there, and all the ACTGs. But what sort of information is put alongside, and you can see on the right-hand side there when it talks about attribution here, it making reference to the place where the specimen is, where the sequence was done, and then people associated with that process. And similarly, it has information about, or at least in this instance here, it has information about where the collection is, or the exact site from where things were taken. You can see latitude and longitude. So I think knowing that this is sort of the standard part, where things are collected from, and then who's been doing the work, but there's nothing that sits in these places about relationships with Indigenous communities. So I'm going to pass on to Aimee to talk a little bit more about the actual data itself.
Thanks, Maui. So this, we understand that not necessarily everybody has some background information in DNA. And Maui's just mentioned there were a whole lot of AGs, T's and C's, and that's really the makeup of DNA. It's just made up of four bases. And from that, there's been a lot of work done to interpret a lot of stuff regarding DNA. So, for example, how different proteins are coded and things like that. So what I've done here, I've just tried to show how do we actually work with the DNA sequences we get back. And before we can upload it into the databases that Maui just showed, we really have to have good quality DNA to be able to do this. And so this is an example from our EnviroLink project that I'm working on. And here I've just shown a CO1 gene region, which is typically used for species ID. And what happens is we get it back from the sequences and we would upload the data file into a program such as this. And I'm just using Genius as an example. So it's quite a commonly used program to look at the DNA sequences once you get them back. And what we can see is that the DNA in the beginning is really messy. So we really have poor quality in the beginning. And then we also have poor quality in the end. And this is really important to basically get rid of, because if we want to upload a high quality sequence to be matched, we don't want this noise in the beginning and the end, because it could influence how the taxonomy is assigned to the different species or sequences. So what we do is we would clean it up. So in this example, I've just done very basic, and we just cut a bit of the beginning and the end off. So Maui, if you can just go to the next slide. And then we get something like this, and it automatically gives us a much better quality sequence that we can then use to look, well, what would the taxonomy, what taxonomy would be assigned to this DNA sequence. So effectively, what species could we find? And so this is the databases are freely available. So we use something called BLAST, which is a program. And what you can do is you can take sequences that are found in GenBank, and you could even run this yourself if you wanted to. So what we do is we take up the cleaned up sequence and we input it into the query sequence section. And then all basically do is you hit this thing called BLAST. And that what it does is it takes the sequence that we've cleaned up and it will run through the entire database it has. So GenBank is a public repository. And what it does, it will take that sequence and it will try to match it to the best sequence possible in its database. And so the output examples, Maui, next slide, please. Is something like this. So once you've hit BLAST, it will take, depending on how good the sequence could match and how quickly it can find a really good match for that sequence. So it could be a few seconds to a couple of minutes. You get a new screen which looks like this. And what it's saying is that, well, the DNA sequence that you gave us, these are the top matches that we could find at species level. And so then typically, there's a whole lot of statistics you can look at to identify, well, how well did this sequence match a species of that we know the DNA sequence of in the database. And just for example, next slide, please. You can see that here we know that because this project is based on voucher specimens, so we have taxonomic experts looking at what species we extract in the DNA from. So we know that this is the exact species that we've put in. So it's a species of mayfly. And then when we ran it through the GenBank database, well, we would actually hope that it would come up with that if it is expected to be in the database. And in this case, it is. So you can see we have a percentage identity match of 98%. So that means that the sequence that we put in matched really well to a sequence that was in GenBank. It's not 100%, but that is definitely a high enough percentage that we would say, yes, this is a species level match. And we would quite confidently, if we didn't know the species, the origin of the DNA that we had put in, we would say, well, yes, this DNA we can identify as this species, so therefore we know it is in that area. But then what happens if we don't actually know, well, if we don't know the species and perhaps it's not in the reference database? Well, in this example, we've put in a different species of mayfly, and we've ran it through BLAST, just like we did with the other one. But this time we get, oh, that box is supposed to be a percentage ID, but we get a really low percentage ID of 83%. And at this level, we wouldn't actually be happy to say, well, this DNA is this species. There would be other programs or software that we could use, and perhaps we could get it to genus level or family level, but we wouldn't know what species that DNA specifically is. And so that's when we are really needing to know the species ID, that's not really good enough. If we're only getting it to family levels, and perhaps there's hundreds of species within that. And so this is really the importance of building up a database. So if we're doing a lot of eDNA work, that's been happening with the councils, then we would like to know, and I'm very confident and be very confident with our results, that the DNA we can put back to an exact species match. So Maui, back to you.
I was going to say, so this is really highlighting the need for a DNA reference database for New Zealand species. And what I want to do is I'm going to stop and ask the panelists, like, why do they think it's important to have a national DNA reference library? Maybe Manpreet, let's start with you, and then go to Libby.
Yeah, just to continue on the conversation around the National DNA Reference Library. And as you had some of those points on that slide as well, Maui, I think we have to think about the issues that are affecting across the country, so we can build a database that allows us to monitor species decline, species losses, impacts of conservation practice. So a wide range of applications that could be supported with a national reference resource. And I think the efforts like the ones that Libby's involved with show that, you know, you can do them at place and with certain groups that are really underrepresented. It's also widespread. New Zealand diversity is quite unique in many ways. And so while we do have access to some information from overseas collections and global databases, it is imperative that we do have New Zealand species represented, because that's the best, most granular information we can use to make better informed environmental decisions. So I think the need for the reference database from a long-term application point is fairly obvious, but how we go about it, I think in New Zealand, is what the key question here is. How we make sure that this resource will be uptaken by not just scientists, but also community members who are really the kaitiaki going forward. I think I believe that New Zealand, with all our resources that we have at our disposal, it's small. The problem is bigger. So we do rely on our communities to be, really proactive in managing the resources, in managing the diversity. And so I think this is a resource that needs to be for everyone. This is a resource that needs to be accessible and done in a way that people want to engage with it. And this is kind of the thinking we've been trying to have in the national project. But again, we're learning all the different aspects. We're learning from the concerns people are raising as well. And I think that's why this is important to keep talking about it and keep bringing more and more viewpoints into this conversation. And I think maybe that's where I can hand off to Libby if you want to say a few points. Libby.
Thanks, Manpreet. Totally agree with everything you've said, and just building on a few points. I think eDNA technology really fills a niche that needs filling in New Zealand. As Manpreet said, resources are stretched, and the feedback we had from Council, and obviously the impetus for the EnviroLink project, is that Council is really stretched in all of their management duties, and they're looking for technologies that will enable them to do their job, but in a way that is efficient and can be done by people who don't always have the specialist taxonomic expertise, which is increasingly a problem around the world. We're losing taxonomy and taxonomists, unfortunately. And it is a slow science, whereas eDNA is a fast science, and it's one that is accessible to the greater masses. So as Manpreet said, the idea is that, yes, Council would potentially use this library that we're creating as a tool, as a resource, but it also would be a way to increase engagement with communities so that those tools and resources become accessible to them as well. So local communities, hapu, iwi, however it's most appropriate, we decide. So there's a real niche for eDNA technologies. The other thing is, I think that the importance of having a national approach, and Manpreet started to talk about this, is that the generation of DNA reference sequences is happening. Many scientists in New Zealand, such as myself, have been doing it for a number of years, and we're creating data that potentially is going to go to waste or not be looked after appropriately if we don't grab this moment now, where there's so much momentum, and provide some guidance and have an open conversation about what is appropriate use of this data, or generation of this data, what is appropriate, how should we store this data, and how should we be using it. And I think that these kōrero we're having now are really timely and really crucial. We need to get to a point where we have a greater understanding, not only within the research community, but also appropriate social and cultural license for this technology, its use, and the data governance, so that we are using the tool in a way that is acceptable and people are happy and feel involved and engaged, which is really important for our biodiversity futures. Like Manpreet said, it's not up to the scientists or the governance agencies anymore. I think it's up to us as individuals, and we want to participate in looking after the well-being of our environment.
Well,
thanks, Maui.
Holden, anything you want to add?
Yeah, I did want to add something. I mean, I take on board what's been said, and of course, there is a need for a national reference library. But what's really important for me is that it is underpinned by Te Tiriti o Waitangi. This is Māori data we're talking about. It's Indigenous data. Hapu and iwi have Indigenous rights and interests in that data under Article 2 of Te Tiriti o Waitangi. We need to figure out how that permeates the governance of how such a library would be managed or governed. So there's that big question there around how we do that. And that's I guess that's what's attracted me to this kaupapa, really.
Well done. Thanks. Hey, so I think it's kind of really interesting. I see there's just a point that's been put in the chat here. From my experience, global libraries have been developed opportunistically. And I think you'd probably say the same thing in the national context based on what Libby's saying, often neglecting regional national needs and specific species. In contrast, natural libraries have typically developed based on these strong focus on closing gaps and maximizing utility of reference samples. So that's an interesting point. There's a couple of questions here, Libby. One was, is the DNA itself also archived? And if so, how, where?
Yeah, so in the case of the EnviroLink project, Auckland Museum, Tamaki Pangahera, has agreed to develop their own, I guess, facilities to have a DNA extraction database. So they've recently moved out of just doing solely specimens, which is, museums typically do, to include tissues. And they're thinking in the context of this project, given the importance and, trying to establish new norms, they're going to also incorporate DNA extracts in their collections. Whether all national collections and facilities have the resource to do that is a question mark. And we haven't yet come to, a conclusion for the Ministry for the Environment funded work, with probably ESR or NIWA would be the appropriate places for those extractions to be held. But they are very, very valuable, I agree, as a resource for reuse, but also just for looking after. They are derived from that biodiversity that we all care about there, that interim between the data and the biodiversity, the specimen, that are somewhat more tangible, I think, as something to look after. But again, this is something that I think should be a national discussion. Is in terms of our nationally significant collections that get given funding by government, should we have one for DNA extractions, or should the funding given to those entities include the resources they would need to facilitate storing those extractions, that actual DNA?
And just one other question before we move on. Some voucher specimens have incorrect IDs. Is there a process to avoid these errors making it onto a molecular library?
Yeah, so we have a whole series of quality control steps that are building on, I guess, best practice internationally. A lot of other people have tread this path, and there's a lot of data out there that is not fit for purpose. It's not being, I guess, ID'd appropriately and whatnot. So Aimee and I can probably speak to that a little bit more, but there are steps in place. I won't bore you with the details now. It's a bit manual, a bit of to and fro. Even the best bioinformaticians would accept that there just needs to be a little bit of manual curation every now and then, a human eye to look it over and sense check it. But yes, that's certainly a concern, and one of the reasons why we are wanting to use specimens from collections that have been ID'd by experts that we know and trust, basically. And that's another reason for having a national approach to a DNA reference database, because we can control, I guess, what specimens are used to develop the sequence library, as opposed to a lot of people who generate reference sequences might go out and catch the fish themselves, and they may not themselves be a taxonomic expert.
Thanks. Okay, so let's go back onto some slides. Am I on the right screen here? Yes. Okay, so one of the things that we were had a look at is what the sorts of questions that arise or are always asked in the context of these kinds of projects. So where is the data held? And so these were just some sort of initial questions which I bounced around the project team members and saying, what, as experts or people working in this area, where might these things be sitting? And so we've seen here the suggestion around bold. Previously, the examples of where the samples were is an example of information that's then being stored in bold, or you get GenBank, or the Aotearoa genomic data repository were suggestions. Who owns or has interest in the data in the library? And so there's always this mix of differences between people thinking about it in the context of public benefit, international interest, that the ideas around ownership are complicated, but we start talking about who should the custodians be, or who should the kaitiaki be in that kind of context as well. Is Indigenous metadata enabled? They said there should be possibilities for metadata columns for relevant information to be added to standard templates. And who would govern any library, and whether that should be a dedicated team, whether that should be experts, it should include mana whenua. There's all sorts of questions that need to be thought about and bounced around. and how will the governance give effect to local hapua niwi? And I think that's something to think about as collections get put together from a whole series of different projects. There might be local relationships around the project, but that doesn't necessarily translate into how things are used as they're all collected together, or how that gets managed or their input gets managed as they're all collected together, and whether the library should be open access or whether there can be restrictive protocols for certain types of data. So just wanted to say that we did a bit of an assessment of some of the data repositories, in part based on ideas that come out of Indigenous data sovereignty, thinking that emerges from the Nagoya Protocol, the key principles for Indigenous data governance, and a new Indigenous metadata communique. And just thinking about some of the publications that are starting to emerge about thinking about how the care principles in particular are being or influencing the way people are thinking about biodiversity science and biodiversity data. And recent articles around Indigenous rights, Indigenous people's rights and data, but also this Indigenous metadata or Indigenous metadata bundle. And I thought there was something there. Anyways, well, we'll send out the link to that bundle. But we looked at these databases and a toolbox. And so the INSDC is the International Nucleotide Sequence Database. That's one of the databases which holds all the DNA sequences. The Earth Biogenome Project, which isn't a database itself, but provides guidance around what sorts of how the data should be stored and maintained across multiple projects around the world. So it's sort of like a bit like a standard setting exercise. Embrave, which is at the Centre for Biodiversity Genomics in Canada, is a place where alongside this, they have Embrave and the Barcode of Life database. There's a European Reference Genome Atlas, which is involved with part of Earth Biogenome Project. The Systematics Collection Database at Manaaki Whenua, where they have all their national collections added to the Indigenous Aotearoa Genomics Data Repository, which has been put together specifically by Nessie, and a Marine Biosecurity Toolbox. And the things that we looked at were these. So these topics down the side, was it obvious from looking at the information, looking at the websites or looking at their policy documents that they're supporting Indigenous governance? Was it clear where the database infrastructure was? Did it have a governance group? What was their policies around data accessibility? Are they promoting consultation with Indigenous people? Is there expectations around anything around Indigenous provenance, or is that enabled within the fields which they provide? Do they have geographic governance? Information of the samples and the species retained? Are they promoting care? Things around metadata standards and Indigenous metadata? What sort of links do they have to other databases and their data sharing and management best practices? Any statements around intellectual property or the Nagoya Protocol? And things around digital rights management. So just where people are using or being explicit that the data provided is in the public domain. So this means it's just able to be reused by anyone, or whether it has a Creative Commons license, and then whether they utilize the local context notices and labels too. And so and just on the right hand side there, you can see these are some of the fields from the metadata bundle, which have been put together that said it's important for Indigenous communities to have information about in the areas of governance, provenance, lands, protocols, and local context. And so just seeing what those look like. And the general reflections based on what we looked at across those databases is that most of the databases are completely open access. A few of them enable the ability to restrict access to the data, whether that's for Indigenous control or any other needs. It's unclear whether there was expectations that the data which they held for others needed to involve consultation with communities, whether that's permits or consent or other things. When governance was talked about in the places where there was something there, it was more about the governance of the platform itself rather than the data or the data sets within it. And most did not have any fields for Indigenous provenance. And while most of the databases were open access, they didn't talk about FAIR, and only the Earth Biogenome Project referred in part to the clear principles for Indigenous data governance. So I think that's where we got to with looking at those, with looking at the databases. So that's what sort of the existing situation is. And so, really today is about trying to start thinking about how could it be different, or what needs to be different in the ways in which the databases are governed and organized. So I think that's our slides for this bit. It's kind of opening it for questions and answers now.
Maui, if I could, there's a really good question there from Claire Adams, just asking about how we've actually gone out and engaged with iwi and hapu communities so that they can input into this kaupapa. The reality is the best in the funding that's available for the contract that we've got. We've tried to reach out to Māori interests and have them engage in these wānanga. One of the clear pieces of further work that needs to happen here is when we do feedback our advice to the government, is that there needs to be a much more extensive level of engagement with hapu and iwi communities. That's coming through really loud and clear for us, I think.
And the other part to it too is that there's need in some ways you need some of the things that we know what they're likely to say, because they've been saying it to us in other contexts through the development of other projects. Yeah. And what we're trying to do is pull together some different sorts of options that they can then say which one best suits their needs. And part of it is also, having a feel for the context and what is then going to be maybe optimal, what's practical in the context of how these things work. So there's definitely also this bit of, what can we do and keep things in a national context versus where things sit in an international. And so there's already lots of things sitting there. So I think we have to think about that component too. Any other pātai or reflections? Well, I knew you'd have a question. Experts in treaty jurisprudence and litigation being involved. There's no one on the project group. Holden, you want to have a crack at that?
I wouldn't pretend to be an expert, that's for sure. But that's another good question, and this is where we need to make it clear to the government that there needs to be a much more meaningful level of engagement with Te Tiriti expertise and hapu and iwi communities, puta noi te motu, So, good point.
I think there is, if we go back to some of the questions asking around who owns the data, or where is this for? In lots of ways, I'm a bit open about this. It sort of gets fudged a bit. So on one hand, we have advocacy around kind of a treaty position, and that data is a taonga. But even with Y262, it's never quite got to the ownership of it, and the ownership of it goes into was it at least in an intellectual property sense, because no one's in a position to do that way. So what are the control elements that allow you to be able to have authority over the way in which the data is used that might be seen to be akin to ownership? And
yeah, so just building on that one too, Maui, I mean, and Phil, you'll be interested in this. I mean, I'll take my Manaaki Whenua hat off for a moment and put my iwi chair hat on for a moment as the chair of Ngāti Maru. From my perspective as the chair of Ngāti Maru, we own that data. That data that's been collected from our rohe, we have an ownership interest in it. We have rights and interests in it. So I guess to that extent, I make no, you know, I'm quite clear. We have ownership rights and interests in the data.
And I think there's there's ways in which I think that can come about in parts in a formal way through the process. And I think this is the bit once you get onto the records. So both you've got the policies that get in place around who gets access to data, and that's sort of a decision around where those things sit. So that's one about access and who gets a chance to have a say around that. And there's the bits around what the actual ownership looks like, and the ownership and what you're wanting to be able to control through the ownership, through the claim of the ownership. And and I think this is your point, Phil, around the litigation, what you can then do if something untoward is done, or someone, you know, misappropriates or uses that data in a way that, you know, you don't agree with. What sort of recourse do you have? And that can only come about by having, in some ways, a legal response to it. Cool. Any other pātai? Oops. No. Is that a no? All right. Okay, Claire, I'm sure you can just jump up and ask your questions. Okay, what do you do about a wide range, open access to closed ownership of Māori opinions? And what if there are a wide range of opinions when multiple iwi claim a particular rohe? Holden, are you going to have a go at that one?
Yeah. There's no simple answer to that one, Claire. I mean, I think, and this is where I think for a kaupapa like a national reference library, there needs to be a national level kind of national level of Indigenous governance across it. So the extent to which you could incorporate, quite aside from the overlapping interests question aspect of your question, there needs to be a national level of governance, and the ability to actually incorporate hapu and iwi perspectives, specific hapu and iwi perspectives into that national governance will be the real kind of determinant of success of the kaupapa, really, I think. I suspect that hasn't answered your question, but it's not an easy question to answer.
Yeah, but I think it does start to, you know, really point towards the fact that there are overlapping interests, and it's like, what are the ways and where are the places where each of those can be accommodated? So how, in a sort of a collection event, can there be ensure that there's an appropriate conversation with local mana whenua, an agreement with that? Any of their wishes, how are their wishes then represented or reflected within the I guess the records that sit in place, and sort of the architecture of the platform, and then the governance of the National Reference Library itself, just as Holden's saying, might be more of a kind of a national, whether it's expert or some more kind of treaty framed governance group, but they're going to set in place the I guess the conditions within which those things can operate. And in some places it might be making decisions on behalf, and in other cases it might be sending the decision making down if that sort of decision making is required, or if and when that is needed. Oh, I see a few things popping through now. Is your view who needs to understand the principles underpinning the kaupapa, and how can that education take place? Education of stakeholders seems to be an important part of this. Panelists, any comments, thoughts? I will just say yes, no, that will be. I think once there's, I mean, part of having the wānanga series and then this webinar too has been about trying to stimulate conversation and thinking around it, what some of those issues might look like. And certainly once you have the framework in place for what it might look like, then there'll be another conversation required just to make sure people are comfortable with that as well. I see a health sector bias, and wanted to know about healer cells and the hurt cause. Can we not use examples such as this? I know, Phil, if you want to talk a little bit about what you're doing in the health space around governance.
Well, kia ora koutou. Yeah, just quickly, in a previous one of these series, we talked about the Rake Ora and Aotearoa Very Own projects. Both of those projects embed Māori leadership as well as governance over the resources being developed, and also include development of those Māori attributes into development of those resources. And Claire Rai, who's here, can probably speak to that, and from a programmer's perspective around how that's been enabled. But yes, the short answer is yes, there are. These, from our perspective anyway, are treaty compliant. And I've discussed these with Holden and Manpreet and others. And there are internationally, there are other models of Indigenous governance, such as the Northern Region Biobank Initiative, involving people like Ngādeen Karaon in British Columbia. And we're not the only communities that have to address the issue of trans-tribalism and how to implement tribal governance or trans-tribal governance over these sorts of resources. So I think there's some learnings to be had, as I put up in the chat, both from what's happening in this country as well as what's happening in other countries too. Kia ora.
Kia ora, thanks, Phil. Got another curly one here from Alex. If I collect and process, for example, aquatic invertebrate samples using standard methods, KickNet, then ID in laboratory, and create a data set of taxa present, would iwi have interest in ownership of that data?
Well, there's two concepts that apply in this space. There's mana whenua and mana moana. And, that mana moana applies to the sea. It's not just the material or information that's collected. It's where it's collected from that creates that connection back to us as hapua niwi. So I guess I would be saying yes.
And so if I think about where we got to with some of the projects we've done with Manaaki Whenua as well, from a whakatohia point of view, there was material which is sitting in Manaaki Whenua, and we say we have an interest in it because it came from Aotearoa. What the nature of that interest is and how that needs to be expressed is then often dependent on a conversation. So what we've said in the first instance is that anything that's collected from Aotearoa has to have a connection, but like we have to be represented in the record, so people know that it came from our place. Like how much, how much further that extends, we haven't had any more sorts of discussions with Manaaki Whenua about that. But it would probably depend on what sorts of activities were being intended.
Yeah, there's a good comment there from Phil, which is kind of a part response to that question. I mean, the principle that certainly we're trying to adhere to is, seek prior informed consent before we do the collecting, because there is always going to be that tribal hapu or iwi interest in Aotearoa. So prior informed consent is a good starting point.
Okay, kia ora. So it's sort of, I think that's where we've got to in terms of the Q&A. And now we're intending to do a couple of breakout sessions. One that's going to think a little bit more about governance, and one that's going to be a bit more around protocols and data management protocols. So Q&A with the speakers. Okay, exploring Māori data governance. It's just important to sort of think about the governance of the repository and the governance of the data sets, and how they and how those things relate to each other. And so in relation to, what does the repository do? What are the sorts of standard operating procedures they put in place? How does the people involved with the governance there inform their approach to data governance? And what sorts of things? I talk about active and passive data governance, at least in the sense of active, when are decisions required to get access to information, and when are passive is just around the sorts of policies that get put in place to shape what can be done or not. And then they also, I think, are involved with informing what sorts of information is required around things that get deposited. So then you get to that governance of the data sets, and with the thinking about active and passive there, is there a sort of authorization process for them? Or is it just that you're reflecting the consent and permissions and things like that sit there? So there are 5 questions for the people in this first breakout. Where should the data be hosted? Who should be a part of the governance group? How will it maintain accountability to iwi and hapu? Should the library be open access? And does different data require different governance? So just really thinking about what kind of protocols then get put in place, and how those are thought about, because it's quite easy to just think about governance and say actually that they've got it, as soon as the governance place there, it's sorted because they're around. But actually, what are the things that they put in place, and what kind of protocols are there that allow you to achieve the aims that people are talking about? And I think it's been pretty clear that there's kind of widespread support for treaty governance, for greater control of communities around sort of the data that they provide. But what can that look like through the systems we put in place in terms of access protocols? What are the bits that might be sitting in a public domain space or available with Creative Commons licenses? What sort of authorization is needed to get access to the other bits of information? Is it the researcher? Is it something around the way they that there's a sort of a data access committee or something like that? And then how does that reflect any kind of use restrictions that get put in place by people that are, as some people said, like the owners of the data or the providers of the data? So they're not necessarily going to be involved in all of the operational decisions, or there might be cases where they are. So how is their consents or permissions? And these things might sit alongside, because it might be a consent from a landowner, but it sits within the rohe of mana whenua, and so different things required. And then what does that mean for what sorts of information might be obscured in different ways? And so this was just some kind of a decision tree that got put together, and was thinking about whether data should be accessible or not. And it sort of works its way down, and you get to the bottom, and you got these three different sorts of options. So one is outside of things being, I guess, fully open access. but you might be restricting a certain type of data. So you could mask the species IDs, but not the locations or the high-res, or publish the high-res habitat maps. You might restrict data that masks the species locations, but not the IDs. Or you might be masking both the species IDs and the locations. And I think the thing here is that you can see that there could be different ways in which bits of information could be managed. Some of them then require some active decision-making by people, and other bits are just a part of the process of how things would work. So I think that's something that informs there. And then what sort of other information might be sitting alongside things as well? So metadata requirements. How do you ensure, even if the information is available, that they can see where it comes from, the Indigenous provenance, that there's been appropriate consultation, or that there's information around the geographic location, and then any attribution requirements, whether that's Creative Commons ones or whether that's labels and licenses. And there's different sorts of expectations that can be put in place by different groups. And so a little bit on the right-hand side, that comes from the European Reference Genome Atlas. And so they've got a metadata manifest that says, what's expected for reference samples. So we've got our next breakout. Here's the questions. Some of them have probably been covered in some ways, but should different data get different treatment? Who should inform use restrictions? What access options could be enabled? What Indigenous metadata should be collected? And what attribution should be required? Oh, kia ora koutou. I just want to say thanks to everyone for hanging in there for the whole 2 hours. Well done. You get to go and have some lunch and things now. But just say it's been really great to have a chance to kind of talk these things through. I think it's provided a bit of direction to the project teams. We'll be able to talk a little bit about it and see where they end up. Like I said, there's upcoming wānanga as a part of that series. Next week there's one, but in October there's one that we'll be talking about Māori data sovereignty and Māori data governance as well. So there'll be some different speakers involved, and hopefully we'll be able to pull together some of the thoughts from this and share that as well too. So thanks for that. And if I can just also just thank all of the panelists for making themselves available. It's been really great. And kick back to you, Holden. He whakakapi ngā kōrero.
Kei te pai, ka tukuna te rākau ki a koe Maui mau hei whakakapi tō tātou hui e hoa.
Yeah, okay, kia ora, kia ora. All right, well, thank you, everyone. Just in closing, kia tau kia tātou katoa, te ata whao tō tātou iriki karaiti, te aroha o te atua me te whiwhinga teitang ki te wairua tapu, ake, Aimee.
Aimee. Kia ora Maui, kia ora tātou.
Kia ora koutou.